So it is July. Thank you all for making it to July and sticking with us. The last time we met was, I believe, January. So it's been quite a few months, and we've made a good amount of progress on the data generation side of OASIS, along with some study design factors that we want to update you all on today. And so as per usual, this is our meeting to just provide consortium-wide updates. So we'll start with some achievements and some updates from the group. And we'll spend a good chunk of time today hearing about our data generation updates, specifically what's been happening at the Broad, because they've been very busy, and give some information about access to the data that has started coming out of the Broad. We'll also spend a little bit of time talking about data analysis planning. I did send a document ahead of time in the calendar invite, and I'll make sure to link it when I resend the notes. But this has the data plan, draft plan for how we're breaking apart the data and putting things into manuscripts and starting to think about analysis. And I know this is a really exciting part for a lot of our industry and consortium members. So please take some time to review that, whether it's during this meeting or afterwards. We'd love to have a discussion about how we can get those of you involved who want to be more, more involved with the analysis. So I'll go ahead and start us with some announcements. This OASIS Consortium is part of the Emerging Systems Toxicology for the Assessment of Risk Committee housed in HESI. And I know some of you are part of the ESTAR committee and some of you are not because of the way this consortium is arranged. But just to give you a little bit of background, every year this committee has a annual meeting. And this year we've decided to break it apart into about like 1.5 to 2 hour meetings for each of our work groups. Those are listed here, although I realized I forgot one because we have 6. But if you've been getting calendar invitations for any of these work groups, you are more than welcome to attend. We would love to have you learn about the other projects that are happening at eStar. And if you want to get more involved, you are always welcome to do so. So I sent out an invitation for the OASIS Consortium on Thursday, November 13th. But as I mentioned, if you're getting emails that have any of these other work groups in it, you are definitely invited to attend, and we hope to see you there. We have a Eurotalks presentation or session that is David and I are chairing together. We have a great lineup of speakers that will be presenting on fundamentals and evolving uses of cell painting and toxicology studies. If you happen to be in Athens at Eurotalks, we would love to see you. So please let us know that you're going to be there, and I'll try to make sure that we are able to connect. And also, if you can attend our session, that would be great. We also are starting to plan for SOT in San Diego in March of 2026. We will have a HESI get-together, potentially an eStar brainstorming session. eStar has a session on PODs, but for OASIS, I'd love to just throw out the idea that if anyone is going to be there and would like to present on behalf of OASIS, we could consider an abstract submission. This is always a really nice way to get people there and talking about the project. So if you have ideas or if you just want to volunteer for helping to put something together, please let me know. Okay. I presented on this last time, but just wanted to give an update on the OASIS compound library, which is being prepared by SPECS, which is part of eMolecules. And David, do you want to give a 2-minute overview?
Yeah, sure. Well, first of all, thank you for allowing me to join. My colleague Johan Tehaus is trying to retire. So I'll be taking over and joining these meetings, and I'll also be your primary contact. So if you have any questions about the set. So yeah, so basically, so we've worked with Ann Carpenter and the Broad on a couple of annotated sets previously, and we were brought in on the OASIS set as well. So Johan and our team in our Netherlands location has been working on this, and what we've done is, we've put together the priority set of 500 compounds. These compounds have been ordered. We're waiting for them to come in. I was trying to get a last minute update on exactly when we'll be able to start shipping these, but it's imminent. And, I would say 4 to 6 weeks most likely. But in short, we're able to provide these sets more or less on a custom basis. So we have sort of standardized the consortium sets at 100 microliters and 100 millimolar concentration for all of these compounds, and then we will plate these basically on demand when we get an order. So that allows us to customize how they're delivered to you. So whether you want a 96 or a 384 well plate, and then we would adjust the prices accordingly for custom sets. So if it's a less, the quantity is less, but and the volume is less, we'll have to make calculations on that. So that's all doable. Feel free to reach out to me directly. At the present time, we have not ordered the compounds for the full set, the 1400 compound set. We have, of course, between the priority set and other compounds we've purchased, we have a portion of these, but we'll wait until we have more of an interest in this before we would move forward with those purchases. So, yeah, in short, these are consortium prices. We will be offering this outside of the consortium, and we've added something to the cost of those for external partners, just our way of being part of the consortium as well. So yeah, I think that's it real quick. And as I said, I'm fully available. Please reach out to me. My email is right here. Be more than happy to chat in more detail.
Great. Thanks so much, David.
Yeah, thank you.
I will distribute this information so you all have it as well. All right. So I will just do my very quick OASIS overview, as these calls tend to have people who have been to every meeting and those who come less regularly or are just interested in learning about the project. So I just want to level set us here. The purpose of OASIS, as you all know, is to gain confidence in the combination of cell painting, transcriptomics, and proteomics for safety assessment using hepatotoxicity as a use case. And so in that context, we spent a good chunk of our time coming up with a list of public and private compounds that have in vivo rat or human data. And within that, as you just heard David kind of talk about, we have a prioritized subset of about 541 compounds that have the rat in vivo data. And this is a chunk as we move forward and as talking about data generation, we'll talk about the full set and we'll talk about the prioritized subset. So that is the difference between those two lists. At the Broad, we have run U2OS, HEPA-RG, and are going to be running rat primary hepatocytes through cell painting and doing cell exposures for transcriptomics and proteomics. And again, we'll talk about this a little bit more in depth as we move forward. And we've had tech partners reach out. The HEP G2 we'll be talking about today. We have Axiom, who we've worked with and have a paper in submission right now for the primary human hepatocytes. And we'll also talk today about the organoids in liver on a chip. And we're really moving through here through data generation and have been thinking a lot about how to analyze the data. Our postdocs and, well, beyond postdocs now have done a really great job at starting the analysis. And so really want to talk today about how to get the consortium involved. On the postdoc front, I think since we all last met in January, we had some updates, but I just wanted to make sure everyone was aware that, OASIS supports one full-time postdoc at the Broad. Previously, this was split between Srijit Seal, who is now at Merck and is involved in OASIS as a Merck employee, and Jessica Ewald, who recently left the Broad to start her own lab at EMBL EBI. And she has also she is continuing to work with OASIS, not in the postdoc role, but definitely in a data analysis role and a mentorship role as she starts to recruit students into her lab. So where that leaves us is that we have Yona coming to the Broad in January of 2026. She will be our full-time postdoc. And in the meantime, we have Runchi Chen, who's at the Broad currently, who will be assisting with analysis kind of in that gap. And he's been really great to work with. So as you see these names come up, just know that they're all doing a great job on OASIS and have been really dedicated to the project, and we're thankful for their help. Another update, we submitted a paper to Cell Systems. That was the primary hepatocyte work with Axiom. Excuse me, this paper is available in preprint, but as I mentioned, has been submitted and will be the first OASIS paper out when accepted. So we'll keep you all posted about that. We've also submitted the OASIS strategy manuscript to Toxicity. This is a really nice overview of the OASIS Consortium. It was written by our steering team. And again, I'll keep you posted when that comes out. Another accomplishment I wanted to just draw your attention to as we talk through today is that we have increasing tech partner contributions. And I think many of them are on the call today, but we have interested partners in helping us with the data generation and data analysis, the advanced liver models, and as we'll talk about in a few minutes, the HepG2 multi-site trial. So just wanted to say thank you to all of those who have shown interest in starting to collaborate with us and make the consortium aware that we are growing and expanding and having a lot of tech partners contribute analysis and generation in kind to the consortium. So really great partnerships there. Okay, on to what I think many of you are excited to hear about, which is our data generation. So what I did here is I tried to outline the data that we are generating throughout the OASIS Consortium. So on the left is all of our cell lines or our models that we're looking at. And on the top column, we have cell painting, transcriptomics, and proteomics. And so within each cell, I tried to highlight who was doing the work and also the amount of compounds that we're running. So as I mentioned, the full set is that 1,500 compounds. That subset is around that 541, although there's definitely some differences within each subset role. So today we'll hear from Jamie at the Broad about the U2OS HEPA-RG and rat primary hepatocytes. And then, as I mentioned, the Axiom paper that worked on the human primary hepatocytes is wrapped up and in submission. And we'll discuss today the study planning that's been going on between the HepG2s and the advanced liver models. So with that, I'd like to turn it over to Dr. Jamie Chi at the Broad. Jamie, do you want to share your slides?
Yep, I can do that.
Okay.
Let's see if it will let me. Oops, window. All right, can you guys see this?
Yeah, it's in not presenter view.
Give me 1 second. All right. Good?
Yeah, looks great.
Okay, great. I can't see you guys now, so if there's any questions, just shout.
Got
it. So I'm Jamie Chia. I have been leading the team that's been generating the data here at the Broad, most predominantly on cell painting and then prepping samples for transcriptomics and proteomics analysis. These slides are very dense, but I did send them to Chrissy so that she can distribute them among all the members, just in case anybody wants sort of the really nitty-gritty details of what we've been doing. But I'm going to kind of fly through these. All right, so as Chrissy said, we were handling 3 cell-based models, the U2OS, the HEPA-RGs, and the rat primary hepatocytes. I'm going to kind of walk through each one and where we are with each data set. So for the U2OS screening, that is completed as of April of 2025. We ran these kind of in 2 batches. All of the commercial and compounds were run first in January, and that was about 144 plates of compounds. And then once we had all the industry compounds in, those were run in April of 2025. So there are two distinct sort of batches of U2OS screening plates. The total was 160 plates of the full subset, the full set of compounds. So that's all done. That data has gone to Ann Carpenter and Beth Simone's group, and they are analyzing that as we speak. For the HEPA RGs, we had done a bunch of assay development, and this cell painting screen is completed as of June of 2025. In this case, we were able to run all the commercial, industry, and the Jump Target 2 plate all at one time. We ran 4 batches back to back to back to back of 41 plates each, so 164 plates, which we cranked out in just about a month. That data is organized and has been sent up to Beth's group for analysis. There's a couple of plates to be redone, but essentially the cell painting arm of the HEPA RG is complete. We did some pilot testing with BioSpider for their transcriptomics transcriptomic arm of analysis. We ran a small pilot run in May or in June of 2025 using HEPA RGs. We sent them a plate that had multiple cell densities, and they ran sort of their controls along these densities, and they showed that they could get good traces and separation across all concentrations. So that allowed us to set up for the actual transcriptomics experiment. One thing I will note is that BioSpider requires that the first 6 wells of column 1 of every assay plate are left empty, because they need that spot to put their in-house controls. So we will lose these data points on each plate, but because the compounds are randomized across each batch, we shouldn't lose too many data points for any one given compound or dose. So because we had the parameters for the transcriptomics, we actually completed that data generation arm of it. In this case, we ran the subset, priority subset of compounds. It was 480 commercial compounds on our hands, and then the 130 or so industry compounds that we received. We ran 3 batches of 16 plates each to total 48 plates. Those 48 plates are currently sitting in our -80 freezer, and they'll be shipping out to BioSpider in the next couple of weeks. We just have to finish the data organization that goes with the actual shipping of the plates. We also ran a couple of pilot runs with Nomic Bio. This is our proteomics partner. We ran a small pilot back in late 2024 using HEPA RGs at 5,000 cells per well. We sent those supernatants to Nomics for analysis using their original core panel. This was to measure about 300 extracellular proteins. That data was returned in mid-January, which is about normally a 2 to 3 week turnaround, and is currently, I believe, in Jess Ewald's and Miriam's hands and being used for a manuscript. When we were ready to send our plates to them, they actually updated us and said they had a newer panel. This is their Omni 1000 panel. It now measures 1,000 extracellular proteins and is more sensitive than their core panel. So we actually sent them a second pilot set of plates. And they're analyzing that now, and they'll be returning it to us. I believe, Chrissy, you can correct me if I'm wrong, that we'll be planning on using the Omni 1000 or some cherry pick of the Omni 1000 for our proteomics analysis on the compound treated cells.
Yep. So depending on the proteomic results, we'll be likely working with Nomic to kind of design a tox reference panel, so a subset of those 1,000 proteins.
So while we did the transcriptomics plates, we actually took the supernatants from the same sort of that same plate and froze those as well. So those are actually sitting in our freezer, ready to ship to Nomic whenever we finish this assay dev and decide on the panel of proteomics. So for our purposes, the HEPA RG proteomics data generation is also complete. So that's very exciting for us. So now our last focus will be on the rat primary cells for cell painting and transcriptomics. We had done a couple of rounds of assay dev in the rat primary cell for rat primary cells. We are actually purchasing ready-to-use plates from Visicol and Hurel rather than trying to grow the cells ourselves. It just makes it a lot easier. So the first time we did assay dev, we actually used the plates from Hurel as they normally send them at 8,000 cells per well in a cold culture with stromal cells. We tested several different lots, and when we cell painted them, what we observed was they're overgrown. They don't grow in a monolayer. The two different, the co-cultures are hard to distinguish from each other. And so we had a conversation with our imaging team here and with Visicol, and we suggested maybe doing another round of assay development to see if we can kind of clean up the imaging side of this, of the cell painting. So we recently did a second round. In this case, we actually had 2 plates that were sent to us. One was a monoculture of hepatocytes only, and the other one was a co-culture of hepatocytes with supporting fibroblasts, and we did them at multiple densities. We cell painted them. They are now sitting with our analysis team. I'm conferring with them to understand from them, sort of from the imaging side, what looks best. And then my plan is to talk to our study design team to understand from a biology side what is best. And then we'll make a decision on how to move forward with the rat primary cells for the actual screening. And the rat primary cells, I will say, we are only doing the priority subset, given that the price per plate is very expensive to purchase these from Visicol. And the final arm of the rat primary cells is the transcriptomics arm. In this case, we'll be partnering with Alethea on their drug-seq protocol. We ran a small pilot in May using HEPA-RG cells. And we actually sent them 3 different conditions. In this case, we sent them condition number 1, which is their sort of normal, we treat the cells, we wash them, we snap freeze them, and we ship them on dry ice. They know they can get transcriptomics data from that. But we also sent them a couple of plates where we had cell painted the plates first, and then asked if they could try to extract transcriptomics data from cell painted plates. The idea here being if we could multitask plates, we'd be actually saving potentially a lot of money on buying the number of plates we would need to run the whole study. So the initial response has been that the normal protocol works fine, but the trying to extract libraries from the fixed plate has been not as successful and requires a fair bit of optimization, and they're hoping to have that sort of optimized by early October. So right now we're in a little bit of a holding pattern here, just to understand how many plates we're really going to need to order before we execute the rat primary hepatocyte drug screen. Once we make that decision, it's going to take us about a month to do the 4 batches of cell painting. And then if we have to do a separate 3 batches of transcriptomics, that'll take it just another few weeks. So we should be able to wrap up all of this in just a couple of months once we sort of finalize these decisions. And that is it on my end.
Thanks, Jamie. Yeah, I think you said you couldn't see us, but there were lots of hands clapping as you presented all the work. And I'll echo and say out loud, we just really appreciate all that your team has done. Thank you for giving them acknowledgements here. I mean, it's been an incredible amount of work. So I'd love to give an opportunity for anyone to ask questions at this point. That was, a lot of data and a lot of good updates. So any questions from the group, you're welcome to put in the chat or raise your hand. Hydrin.
Yeah, just a short question. So for the red primaries, you choose to use the URL source and not, so there was no Lonza real red primary possibility, I guess. I think it's so. Yeah,
we talked to Lonza about actually making fresh plates, but the logistics of it is very hard. The nice thing about the Visicol plates is that we actually teamed up with Lonza, and we're using the same batch of rat donors for the entirety of them. So we locked down a lot for us. So this way we had a little more control than sort of batch to batch variation as well.
That's true. Okay, thanks.
There's a question from Johnny. Was there a plate quality control metric assessed for the cell painting in HEPA-RGs?
That would be an Aaron question. We had jump controls and we had an entire jump MOA plate in there, so I suspect that's what's being used for quality controls.
Thanks. Jihye?
Hi, my name is Jihye. I think this is first time joined this meeting. So yeah, nice overview of that. Yeah, I can understand that, and it was very helpful. And then Chris sent the recently for the some paper plan too. This is very helpful to understand that overall. So my question is that I think Chris and Connie, we met recently for the new study design for that. So Jamie, so we are going to use advanced liver models like liver on chip with human hepatocyte, primary hepatocyte. And then I'm just wondering that in your slide, so you are mentioned that that I cannot remember the exact cell line or cells, but you mentioned that it was a monolayer. So you are trying to the new round, something like that, because I want to clarify that to our model is that not the monolayer. So we are sitting in the embedding with the ECM and then seed into the chip. So it is not the monolayer, but we are our cell painting team. We already have a test. Some MitoTracker is a very good result, and then some of them is still testing that. But that should be issue for the planning for the study.
So
Like
is the
non-monolayer,
We just had Visigal try it because we were having such problems with their co-culture system to get clean images. Like they just grow as these giant clumps, and we could not figure out, like we can't get a clean plane, right? So the original way that the rat hepatocytes are grown, sort of the standard way they do it, is they co-culture and then they wait a week. So I think there's a combination of the co-culturing and then the week-long incubation before we use them that's causing the sort of clumping together. So in the newer system, we had them try either just a monoculture, or they put the hepatocytes down first, and then the next day they put the stromal cells, and then we use them like 2 days later. So they don't have time to really clump on top of each other. So that's what our imaging team is analyzing now, is how those images look from the cell painting point of view. But then I guess we'll have to ask people who know better than I do about these models, is a monoculture model representative? Should we be using the co-culture? We're going to have to find that happy balance between what works experimentally and what works biologically. So I think
so I think we're gonna meet next meeting with our cell painting expertise and then broaden cell painting people. And I think Chris, you mentioned that, so maybe we can discuss about that. Okay, thank you.
Yeah, thanks, Jihai. Any other questions? Joe.
Hey, Jamie. Hey, nice presentation and overall view of the study design. On the co-culture model, I know you use the HEREL model. I too have used this many years in the past with some challenges for imaging. I did find that the HepatoPack model from Peppergen originally, which is BioIVT now, actually works quite well. And so you still have the stromal cell monolayer, but you have islands or colonies of the hepatocytes that are much easier to image. Have you looked at that at all? Have you tried to look at that model?
No, I had no idea that model existed. We went with HEREL because it was recommended by some of the other consortium members here very early on, and so we had reached out to them.
Yeah, it works well in other toxicological studies, and then we found this similar things definitely for enzymes, etc. But for imaging, even when we're at the Hamner, we had some challenges, with that particular model imaging it. But I can share some information about the HepatoPack model, which was developed, by the way, at the Broad, I believe. So.
Oh, okay. Yeah. That's good to know.
Great, thanks, Joe. All right. I don't see any other hands. Falcon, I did see your question in the chat. It looks like Connie may have answered it, but I'm happy to just put up this slide again. So just to kind of clarify here, the full set of compounds was tested in U2OS and HEPA RGs for cell painting, but it is a subset in the rat primary hepatocytes because, as Jamie mentioned, it is so very expensive to run. So we're going to run that prioritized subset through. That's the plan, at least for now, as we kind of figure out costs and things like that. And then pretty much everything else is a subset, just given, how expensive things are and what people were able to offer in kind. So, yep, I'm happy to share these slides and you all can look at it. I have any other questions? Yep.
Yeah, just one related question. The subset that you're doing in rat primary hepatocytes, I think you clarified that it's the drugs that are known to cause problems in rats specifically. Is that also true for the HEPA RGs, that the subset used there, it's not the rat drugs, it's the more human drugs? You see what I'm asking?
Yes. So the prioritized subset is the same across all. So anytime it says subset, we kind of have that same prioritized list. Those are all compounds. Yeah. So those are all compounds that had rat in vivo data. It doesn't mean that they were positive, but we have the biggest breadth of information.
Okay. So we have the rat data, even though we're running them in HEPA RGs, and the idea. Yeah. And okay, great.
Correct. And a lot of those compounds do have the DILI human binary yes/no liver toxic data, but they just don't have more than that. So that's why we went with the rat data.
Yep. I understand that the rat data is much more rich and complete than the human data, even when we do have it.
Yep. Thanks, Anne. Okay. If there are no other questions, I'll move on. So as Jamie presented, we have data, and I know some of you are excited to get to work with that as consortium members. And so I should just clarify now that I'm talking to the consortium members, that you all have access to the cell painting data that is from the Broad. And so I have a document here. I'll send this out, but basically you will have to access repositories through the Broad. There's some instructions for a form you can fill out. But again, just wanted to mention that all of this data will be public and open data when we publish the first publication, which, we probably still have, I would guess, 6 to 12 months on, if, depending on, probably longer, depending on how long sometimes publication can take. So for the consortium members, cell painting data from the Broad is available, but will be at some point available to all members of the public. Okay, I'd like to move on now to 2 feature projects. As we heard, most of the core OASIS project has been completed or is underway with Jamie and her team, and then all of our partners on transcriptomics and proteomics. One big part of OASIS that we took some time starting to think about was how to incorporate our advanced liver model vendors and explore OASIS compounds in cell painting in these models. And so really the purpose of this experiment is to evaluate, can these advanced liver models detect liver toxicity using cell painting? So some of them have done cell painting before. I think this is a newer technique for them. So really, can we use cell painting to understand toxicity? Specifically, we have thought a lot about what compounds we want to select. And given that a lot of these models are expensive and timely, we'll be selecting a smaller number of compounds, probably just 10 to 20, depending on the vendor's ability. And we would like to select compounds which we saw that the HEPA RGs or relevant cell type failed to detect the liver toxicity if the compound is liver toxic. And so we would select these false negatives, run them through the advanced liver model systems, and we may, depending on the model, we may be selecting different compounds per each model. And so the purpose of this is not to compare vendors to each other, it's to compare the model to the other cell types that we've run at the Broad and other places. So the goal with this is to get pilot studies running soon. The purpose for this would be to troubleshoot the cell painting protocol, and then also to understand, maybe to Jihei's earlier question, like how well can we use cell painting in these 3D models? You know, how difficult is it to capture these images and get clear feature extraction? So this pilot will be 2 positive controls and a DMSO control. And the main studies would be, as I mentioned, maybe closer to 10 to 15 interesting compounds that were selected during our data analysis. So I can stop there and open it for any questions or comments. Oh, thanks, Scott, I thought you might be on the call, but I didn't want to call you out. But Scott's here from BioIVT. They're a vendor interested in working with us, and yeah, would be great to have some more information. Okay. So for the advanced liver models vendors that are on the call, I think Jihei alluded a lot of the next step is to have these meetings as small teams with the cell painting team from the vendor, and then also from the Broad and the data analysis team to start troubleshooting and figuring out the nitty-gritty details on these pilot experiments. All right. Then the next featured project that I wanted to talk through has come up because we've had a few individual conversations with OASIS partners or those who had heard about the consortium who had the capability to run HepG2 cells. And so HepG2 cells had come up as a potential cell line for the OASIS consortium, and just through a series of discussions, we had to exclude it based off of resourcing and thinking that HEPA-RG was a good fit for what we were looking to explore. But definitely some partners had expressed interest in running HepG or looking at the OASIS compounds and HepG2s. So because we had a few potential partners, and I listed them here in the blue box, we had a meeting with them to propose a multi-site trial. So we had I hosted a call with the collaborators. We've had 2 calls at this point to kind of set up initial parameters. And EU OpenScreen specifically has started to procure the compounds and will be moving forward. So they're purchasing from that prioritized subset again. I think they ended up with about 440 compounds, and will be running the OASIS compounds through the HepG2 model that they're verifying as part of their consortium study. The Institute Pasteur Korea, led by David Shum, is also very interested, along with Phaneros, which is Jordy's company that came out of Uppsala University. And so we're working together to see if we can kind of have a nice multi-site trial. I'd also say now, if anyone is interested in contributing to this study, we would be happy to add other sites. So if it's something you're interested in, please let me know. For the study design, just to put out there that we're not looking for a full validation trial. That would be a little beyond our scope at this point. So what we're doing is we're standardizing the test compounds. We're standardizing a defined panel of control compounds along with the time point and exposure. However, things that are different at sites include their protocols. So we would like each site to run their cell painting according to the way they've perfected it, so that we can really see if we're seeing the same results at sites that use different protocols, different equipment, different layouts and batching. So not a validation trial, but definitely an interesting opportunity to see if cell painting has similar results across sites. Are there any questions or comments? Oh, I see a volunteer. Okay, Joe, I'll reach out to you. Oh, and I should mention here too, one more slide on this. To evaluate the sites, we would be anchoring to a control of those reference chemicals, as I mentioned, establishing in vitro points of departure, and then seeing if each site classifies each compound as, active or inactive. And then we can do some visualization comparison of the phenotypic responses. Okay, any questions? Great. Okay, now we're going to move into our data analysis plans. So as I mentioned, I sent out a document in the calendar invite, and this is the OASIS Data and Manuscript Division draft plan. And so this was initially drafted by Anne and Shantanu and the postdocs at the Broad in order to start understanding, who is taking leadership of different projects and making sure that we had kind of all of these different ideas down on paper. So I can share the document now just to give you a little bit of a gist if you don't have it up on your own computer. But the rows and greens are agreed upon or finished. So as I mentioned, that Axiom primary human hepatocyte paper is out. Rows in gray are papers with a solid plan, and those are ones that, in probably 1B and 1C here, those are our postdocs. We want to make sure that they get a lot of credit and experience working through the data, so they'll be leading those projects. So there's definitely some more in gray here as we started to talk through who might be leading. And then all of the ones in white are just ideas that different people have come up with, and we tried to categorize them here by either methods papers or papers publishing new data. So the purpose of this document is for you all to look over it, to let us know if there are any places that there are questions that we're missing that you would like to see answered, if there are areas that you want to participate or someone from your organization wants to participate. I think I wrote it here, but we are definitely inclusive of any who want to participate. So this is not supposed to say, we have it all outlined. This is a consortium project. Anyone who wants to be involved can be involved, and we encourage that as well. Okay. So on that note, what I'd like to do is just give you a quick overview of what our data analysis is going to look like over the next few months so that you are able to participate as interested. So on August 19th, this meeting is already scheduled. We'll be having a data analysis presentation by Miriam. She's a PhD student working with David Broquier, and she has been also has been mentored by Jess. And so she'll be presenting the results from that nomic pilot experiment, as Jamie had mentioned earlier. And the full consortium is invited along with, nomic to give us kind of an overview of the study design and then work through the analysis of those results. Hi, Jen.
I have a question. So this is more about also always in conjunction with manuscript. What about the data from the in-life studies from the companies? How you still sitting on them and have not yet, submitted them because we don't know how to submit? Will we have some kind of meeting on that? Or
Yep.
Did we already schedule that? Sorry, maybe I forgot about it.
No, that's okay. So the plan so Hydron's question is that the industries that contributed compounds have in vivo data that needs to be part of this full experiment. And our goal kind of internally was to have a draft of the template that we need filled out by the end of the summer, and then we'll get that out shortly, I guess by the end of the summer as well. So that way we could have all the data collected by the end of 2026. I'm sorry, 2025. Sorry, December 2025.
And the template may be very different than the data we have because we mostly have them in the SAND format, which is usually what we usually use for upload to authorities. Would there be any option to discuss that also, or
Yeah, I would love that. Hydron, if you and I can loop in the others who contributed compounds, I think that would be really helpful to have a discussion of what is feasible. That would help our group tremendously.
Mm-hmm.
Yeah, I was just going to say, Christy, I think we had talked about before, and I don't think Jessica LaRocca's on, but like having her, David Hydron, and the other folks, I think GSK and a few other companies that contributed on a call
Yeah.
to say, here's what we're proposing, what's
Yeah.
doable might be really useful instead of
Yeah.
going out. Yeah.
Yeah. I think that's a good idea. Thanks, Connie.
Yeah. Yep, thanks, Hydron, for that suggestion. I can get that scheduled sooner rather than later so we can have that conversation before the template.
Mm-hmm. Yeah. Okay, thanks.
Okay. To continue on with the data analysis, so we're planning to launch a data analysis work stream. We've had a few internal meetings, or I'm sorry, small team meetings, but we, as I mentioned, want to get many of you involved. So the goal for the next meeting of that group, which should be the end of August or beginning of September, depending on schedules, will be for Jess to present this, overall OASIS vision in the context of data analysis. So beyond what I'm discussing today, but really kind of how she's thought about how to break down all of these really interesting questions from OASIS. And then start with kind of an outline of paper 1B, which is outlined in that document I sent out, which is kind of the next big, all the cell painting data with those in vivo outcomes. So the full consortium will be invited to that. And then after that, the plan is to have each manuscript, as outlined on that document, meet as needed, likely once a month, to discuss specific data analysis questions related to that manuscript. It would be quite overwhelming to have all of the questions meet in one meeting. So we decided to kind of divide it by each of those manuscripts. And happy to invite whoever wants to attend and participate in those data analysis meetings and contribute to papers. Okay. That is what I have on the data analysis plan, but I'm happy to take any questions or comments. All right. That brings us to our closing today. Our plans for the next 6 months are to complete the cell painting, get the transcriptomics and proteomics data samples to those vendors and start the data generation. Whether the transcriptomics and proteomics are done by December is not sure right now, but we'll kind of see how the rat primary cells go. To develop our data analysis pipelines and have meetings as necessary, and continue to share all of our work with you in the scientific community. So with that, I will distribute the data access form and OASIS manuscript division form again to the consortium members. And if you all can review that, send me emails with who on your team wants to participate, or if you have ideas for other manuscripts, if you're interested in leading, anything like that, please reach out. Are there any other questions for today?
Just really exciting work in the chat. I don't know how much you're seeing, Chrissy, but I think we'll have some follow-up for the HepG2s and some of the writing for the paper, which is exciting.
Wonderful. Yep. Thanks for pulling that to my attention. I'll look through the chat and make sure I have everything recorded. Great. Thanks, everyone. I appreciate your time today, and we will have a meeting summary and recording out to you shortly. Have a great rest of your day. Bye, everyone.
Thanks, everyone. Thanks, Chrissy.
Thank you.
Thank you.
Thanks a lot.